Greetings from Cyprus, everybody. It is indeed an extreme honor for me to deliver this keynote address, especially under the unprecedented conditions we are experiencing nowadays. I wholeheartedly thank the organizers for this unique opportunity. And what I shall try to share with you in the next 40 minutes or so is an overview of the latest advances in molecular communication, and in particular Bionanomachines and nanonetworks under the control of interfaces emerging as diagnostic and therapeutic tools with emphasis on brain malignancies, the most detrimental human cancer. Some 150 years ago, Claude Bernard, a renowned French physiologist at the time, defined medicine as a fragile art applied on solid sciences. I literally cannot think of any better example than the implementation of molecular communication concepts and technologies in the management of brain malignancies, which are the epitome of complexity in human diseases. Nanotechnology, as a discipline, was born at the monumental lecture of Richard Feynman. There is plenty of room at the bottom, where he described atomic scale machines and the vision that these machines can realistically function simultaneously. However, he acknowledged that this cannot happen because we, the scientists, are too big. This was, in fact, the first promise for the transformational potential that nanotechnology was going to have in the years to come. The tiny machines would be nothing else but nature's biomolecules in assemblies. So nanobiotechnology and nanomedicine came to the fore with Isaac Asimov's fantastic voyage, where 4 men and 1 woman in a jet submarine navigated through a bloodstream of a man targeting a clot in the brain. What seemed to be fiction science at the time was in fact the first conception of mobile bio-nanomachines. According to my Greek history teacher, if you do not know where you are coming from, you can never know where you're heading to. In order to identify current challenges in nanobiotechnology, we have to undertake a path through history. Early on, scientists were confronted with a series of challenges associated with the acknowledgement that this new technology was defined by 2 divergent systems: biological systems, which are fundamentally wet and organic, whereas most nanofabricated systems are hydrophobic and made of inorganic materials. Therefore, actual progress in this field was made possible only when the initial seminal advances of each contributory field came to fruition. In early 1990s, the challenge was how to physically couple the 2 divergent systems, since biomolecule material interaction is the sine qua non in nanobiotechnology. Experiments were undertaken to specifically couple biology with nanofabricated devices and tools. In 2000s, the challenge was how to circumvent the divergent systems by implementing nanomedicine artificial bio-nanomachines, such as common nanoparticles. By early 2000s, scientists began investigating how to integrate artificial bio-nanomachines into nanonetworks and nanodevices. Being at the dawn of 2020s, the challenge we are now facing is to realize the great promise in disease diagnosis and therapy with nano-device control. It is said that history is repeated, and molecular communication do verify this. Now, 2 divergent systems define a new information and communication paradigm. As you all know very well, computers and networks are simple, fast, and tough. Whereas, and believe me, I can confirm this, biological materials and entities are complex, slow responsive, delegate, and self-determined. Actual progress in this field, molecular communication, began when conceptual similarity was identified between seemingly very different networks in biological communications, or bio-nanomachines. In early 2000s, the challenge was how to map biological communication key concepts, such as information sources, senders, receivers, information carriers, encoding, decoding, and information propagation in networks. By early 2000s, information theoretic work drives molecular communication to a scientific discipline. And shortly afterwards, significant results in physical layer issues were reported. The challenge at the dawn of 2020 is now to realize the great promise of Richard Feynman in disease diagnosis and therapy with bio-nanomachines and nanonetworks. The first milestone on this pathway has been the presentation of a complete molecular communication paradigm by the Suda team at the University of California at Irvine in 2005, where senders and receivers communicate with chemical signals and molecules. In an excellent retrospective review, members of the molecular communication founding team summarize key milestones that have had notable impact on the evolution of the molecular communication field, both at the conceptual and experimental level. I would like to point out those with indirect impact on the work that I tend to share with you today. These include the proposition of the Internet of Nanothings in 2010, the first research with specific application in biomedicine, such as drug delivery in 2013, then development of the first external interfaces in 2014, and at the end of this exponential phase, two critical artificial cell milestones. Then demonstration of the first experimental testbed with engineered artificial cells, and the proposition of the Internet of Bio-nanomachines. The launch of FedCSA Circle, a concerted action funded by European Commission, which was the first molecular communication activity funded by European Commission, could be considered as an acknowledgement of the high potential of this field. There is absolutely no doubt that the epitome of convergence of nanobiotechnology with molecular communication resides in their application in the management of malignant tumors. In 2014, the Russia team reported a communication model between nanoparticles and engineered tumor in vivo, integrating nanomedicine, tissue engineering, and communication. It was shown that the signaling or sender module, which is here blue, broadcasts the tumor location to the receiving module, red, in the bloodstream for amplified tumor targeting. Only 2 years later, the Felicenti team reported an integrated model of drug delivery. An actuator nanomode is in charge of delivering drugs, which here is shown as red particles. A monitor or control mode is in charge of assessing the effect of drugs on the target by measuring specific tumor quantities, which in practice are disease biomarkers released by the tumor. Here are shown as green particles. Finally, controlling the emission rate of the drug release via feedback signals, shown here as blue particles. It was around the same time when the mostly revolutionary conceptual framework of the externally controllable molecular communication was presented, interfacing bio-nanomachines with the external environment and bringing new challenges to the scientific community. The first comprehensive response to the surge of in-messaging and out-messaging interfaces was the application of optogenetics, which means gene regulation via optical signals, for localized drug delivery in the brain by the European Commission-funded project StarTest. In search of bio-nanomachine to bio-nanomachine interfaces has led to the identification of emerging roles for synthetic biology and nanobiotechnology, and new approaches in implantable cells research. Ultimately, the need to decipher molecular mechanisms deep in the human body, which is directly related to brain tumors, is leading to new approaches in wearable device research and promises to take brain-machine interfaces to a new level where computing systems could detect, localize, and control molecular activities. This very simple graph, which discloses output in molecular communication in terms of peer-reviewed publications, delivers some very interesting messages. After an exponential growth in the late 2000s up to mid-2010s, the field seems to have reached a plateau in the late 2010s, reflecting the need of keys that will unlock new breakthroughs. These include the balance between theoretical and empirical research, the need for applications and reaching out to wider disciplines, and the expansion and growth across disciplines such as physics, mathematics, and medicine. This deceleration of scientific outputs has also defined open research challenges, including the establishment of a standard architecture and simulation framework of molecular communication, the design of communication protocols for value-added higher layer functionalities, and of external interfaces to interconnect with the internal molecular communication environment, which is an enabling technology that helps realize the Internet of Bio-nanomachines. Also, the deciphering and endorsement of collective behavior of bio-nanomachines, and finally, learning from evolutionary and adaptive nature of biological systems, implementing biosimilar molecular communication, and of course, obtaining deep insights and understanding of biological processes. So if Richard Feynman was here with us today, he would certainly say that yes, there is plenty of room at the bottom for molecular communications. Extending the roadmap set by Nakano and Suda, we come across two milestones highlighting acknowledgement and rewarding of bio-nanomachine and nanonetwork research that allow therapeutic translation of models and concepts. The Nobel Prize in Chemistry in 2016 for the design and synthesis of molecular machines, and the launch of Gladiator Fed Open, an international trans-disciplinary integrated clinically applicable approach of autonomous externally controllable nanonetworks with bio-nano device interfaces in 2019. Glioblastoma multiforme, the most detrimental human cancer, is widely recognized as a model of wireless bio-nanomachine communications. It can be characterized as the convergence of complexity, stochasticity, heterogeneity, dynamics, and multi-scale natures that together characterize molecular communication networks. For those of you not familiar with the molecular mechanism of cancer, a tumor starts when a multipotent cell we call cancer stem cell, and in this case we call glioma stem cell, which carries multiple mutations that can drive transformation to malignancy, start progressing. This progression can be either transition to a transformation to a new phenotype, new characteristics, of which collectively make the characteristics of cancer. And this function we call GO, or proliferation and self-renewal, maintaining the same multipotent phenotype in a function we call growth. Glioma stem cells are those cells which survive surgery, radiation therapy, and chemotherapy, and under certain conditions, they repopulate and form tumor recurrence. So cell-cell interaction and microenvironmental factors play important roles in regulating the phenotypic switching and maintenance of distinct subpopulations of cell bio-nanomachines as either GO phenotype or grow cells and recurrent tumors. So existing models of glioblastoma growth nowadays are based on the pattern of spreading at the population level, and therefore there is an urgent need of models enabling the study of how cell bio-nanomachines interact as they spread. To meet this compelling need, it is highly imperative that us, molecular oncologists, start talking wireless and start translating tumor evolution to the concepts of molecular communication. Environmental cues in the tumor extracellular matrix, the matrix around the cells, present especially graded doses built in multiple modes of communication. The sensitivity to spatial gradients of solid chemical factors and ligands is known as chemotaxis. The sensitivity to gradients of physicochemical properties of cell adhesion substratum, very important in tumor spreading and metastasis, is known as haptotaxis and is based on graded surface of the surface-bound extracellular matrix products. Going a step further, topotaxis is the direct migration guided by the gradient of the nanoscale topographic features in the tumor's microenvironment. It reflects the outcome of the material properties of cells and their surrounding extracellular matrix. And ultimately, coming to more mechanical properties, urotaxis mode is the sensitivity to gradients of substratum, stiffness, and rigidity, denoting the presence of mechanochemical cues, and is associated with adhesion substrata. We therefore require models of recapitulating the three-dimensional topographic complexity at multiple scales of size and organization. In a recent joint undertaking with the Nagano group in Japan, we studied growing Bionanomachine networks and their applications to glioblastoma evolution and progression within these modes of communication. We first developed a growing Bionanomachine network model that captures the dynamics of glioblastoma stem cells. And here we present the simulation of the spatial distribution of proliferative Bionanomachines presented by the black dot and invasive ones presented by the red dot. Two Bionanomachine subpopulations with differential migrating potential form a growing networks. Aptotaxis and chemotaxis gradients of molecules result to attractive or repulsive forces. And proliferative Bionanomachines stay closer together to form a primary cluster. And probabilistically, they switch to invasive Bionanomachines and migrate radially from the primary cluster. Under certain conditions over time, invasive Bionanomachines switch back to proliferative ones to form secondary clusters. So ultimately, proliferation and invasion labor is divided to form a large-scale network collectively. In a wet lab experiment of human glioblastoma cells in a two-dimensional cluster, we show that the theoretical and experimental models of glioblastoma networks evolution and progression are greatly aligned. And this leads to the conclusion that targeting the molecular communication network and microenvironment can be high promise for future targeted therapies. Enjoy. You can see in basic time, irregularly shaped, spheroid cells, which do not follow attractive forces, but only repulsive and aggregate in three-dimensional structures here at the center. Yes, the theoretical and experimental models of glioblastoma networks, evolution and progression are highly aligned. So targeting this molecular communication network gives very high promise for future therapies, and this is going to be our immediate next work. And I hope we have the opportunity to share this, this new work in the future. The complexity of chemotaxis and comotaxis gradient, as we described them before, as communication modes, are shown in the interaction between glioblastoma stem cells and another subpopulation of early glioblastoma oncogenesis, the so-called glioma endothelial cells, which form the lining of new vessels, functioning in a dynamic interplay in a network of mobile bio-nanomachines. Tumor vessels can be recruited from glioma stem cells, and at the same time, glioma endothelial cells have direct effect on distinct subpopulations of glioblastoma cells, orchestrating the activation of migratory, invasive, or self-renewal potential of these glioma stem cells. In turn, glioma stem cells give rise to functional endothelial cells, and in a complementary network, remote progenitors from bone marrow generate glioma endothelial cells. Now, disrupting this network can become a central therapeutic challenge in glioblastoma management. Glioma endothelial cells and glioma stem cells are organized into a unique structure, the so-called glioblastoma perivascular niche. The perivascular niche is of major clinical significance, as it is here where the tumor recurrence arises. In terms of bio-inspired information and communication technologies, this tumor structural unit is expected to attract great attention as a prototype later follower-based mobile molecular communication network. And this is because physical association and release of soluble factors via cell-cell interactions are augmented and amplified when interacting cells are in close proximity. In order to functionally maintain cell renewal, the glioma stem cells require several autocrine and paracrine factors that are released in the neighborhood of the perivascular niche. And the perivascular niche does allow access to increased localized concentration of these soluble factors secreted by glioma endothelial cells. Furthermore, the physical proximity of glioma stem cells and glioma endothelial cells is a key driver to tumor growth. We can therefore say that the perivascular niche can become a hub for convergence of multiple phenotypes, including self-renewal, maintenance, angiogenesis, and proliferation. Now, distinct phenotypes of glioma stem cells, we can see here, can either migrate across gradients of adhesion molecules and be tethered to the niche by haptotaxis, or exposed to chemotaxis gradients of soluble factors resulting to a prototype system of natural bionanomachines. And deeper insight into the perivascular niche reveals a prototype model of application layer and a series of bio-inspired test beds of functionalities for molecular communication applications. Support at the physical layer is achieved by glioma stem cells that serve as mobile bionanomachines under the chemotactic control of detecting concentrations of growth factors and migration along gradients of laminin, which is a natural polymer found in the basement membrane of the vessel. Structure for formation is an overarching functionality attributed to the spatial relationship of bionanomachines within these three-dimensional units, building a reaction diffusion model based on activators and inhibitors. Basic functionalities now can entail bionanomachine location and control, where, for example, interleukin-8, cytokines are molecules which regulate the mobility of cells, serves as attractor for glioma endothelial cells. Microenvironmental Q monitoring with emphasis on monitoring of molecular communication environment, and the tethering of glioma stem cells to the membrane via complexes of integrin and adhesion molecule with laminin, and ultimately control of molecule concentration such as the delivery of growth factors. The strong message we can receive from the past three slides is that in order to find the most appropriate nanonetwork validation model, it is essential to recapitulate the in vivo microenvironment. In particular, to validate human molecular communication theoretical models, we do need to build basic test beds enabling quantification of model parameters, but most importantly, need to first answer what do we want to measure, how we want to define, and how we want to analyze molecular signal metrics. So for successful disease diagnostics and therapeutics, we need detection and quantification of appropriate marker for disease. This is the sine qua non of oncology. We need deep knowledge of the spatial temporal and disease evolution. And we have to define, design, and synthesize suitable bionanomachines and nanonetworks. Coming to tumor pharmacology, which is a central discipline in therapeutics, we realized that it needs to be transformed to a new discipline where drug development testing models are directed by the concepts of molecular communication. In this context, therapeutically engineered cells as natural nanocarriers can be developed as prototype bionanomachines and ultimately recapitulate the in vivo conditions, applying the modes we have already discussed for molecular communication. Therefore, the challenge we are now facing for swift translation from the theoretical model to the clinic is how to translate the layered architectures to multi-layered tumor models. Three-dimensional tumor models comprise a significant technological advancement of the last decade and serve as valuable test beds in molecular communication because they combine tissue engineering, cancer biology into a molecular communication environment. Three-dimensional cancer models is the outcome of the implementation of a diversity of methods such as three-dimensional spheroids of cells, bioprinting, assembly of biomolecule, and biocompatible materials such as hydrogels, scaffolds, or even their combination into hybrid systems like this one of our own work hosting glioblastoma stem cells, and provide unprecedented backgrounds for the development of neurotaxis, biophysical factors, aptotaxis and topotaxis with microenvironmental parameters, and chemotaxis via gradient concentrations of different chemical factors. So, in an effort to take a big leap forward and take molecular communication to a drastically new level, 7 teams across Europe and Far East, led by the University of Cyprus and Cyprus-based Nanobiotechnology SME, joined visions and forces towards the next generation theranostics of brain pathologies with autonomous, externally controllable nanonetworks. The Gladiator Fatal Pen is the first international, interdisciplinary, far-sighted, integrated research approach in externally controllable molecular communications and has used the circle TED-CSA as a strong networking foundation. It can be considered as a paradigm shifting between disciplinary taking, introducing a working prototype of a complete, autonomous, and clinically applicable network-based molecular communication platform. An overarching science to technology breakthrough in Gladiator is the study of glioblastoma as integrated interactive networks of cell and exosomal bio-nanomachines in the tumor microenvironment. Exosomes and extracellular vesicles are natural nanostructures of cell membrane encapsulating biomolecules such as RNA, DNA, protein, and they are released by a function known as exocytosis and uptaken by other cells via endocytosis. Exosomes can therefore build prototype nanonetworks, communicating information among cell bio-nanomachine, such as glioma stem cells, glioma endothelial cells, astrocytes, and tumor immune cells, such as the tumor-associated macrophages or T cells, maintaining integrated interactive networks in the tumor microenvironment. In Gladiator, exosomes as natural nanoparticles and bio-nanomachines can be subjects to external control of radiofrequency, for example, serving as the building blocks of a diffusion channel model where neural stem cells engineered to produce reprogramming, which means therapeutic, changing the function of a gene. This is reprogramming. Exosomes serve as transmitters and can release and diffuse to the brain extracellular matrix, which is here the communication channel, other exosomes which are uptaken by glioma stem cells serving as receivers and maintain bio-nanomachine to bio-nanomachine interfaces. So what Gladiator proposes is literally brain tumor management as a fully autonomous, externally controllable molecular communication nanonetwork, where orthotopic tumor foci of glioma tumor cells and glioma stem cells, implanted organoids of therapeutically engineered neural stem cells, and implanted sensors of optically active cells serve as communication network blocks building 3 distinct channels, and where the optical signals generated at the sensor cells by tumor release monitoring exosomes serve as the out messaging interface. An external device translates this message to the radiofrequency signal that regulates the genetic stimulation of growth molecules found in the reprogramming cells. This externally controllable molecular communication network and individual components are integrated into a clinically applicable working prototype of Bio-Nanomachine Diagnostics and Nanonetwork Therapeutics. The validating in vivo model comprises a brain with orthotopically planted tumors of both gold glioma cells and grow glioma stem cells, subpopulations, and autologous organoid, A here, of reprogramming cells releasing the rationally designed exosomes, the therapeutic exosomes, the implantable hybrid sensors containing cell-based sensors and electronics, with the cell-based sensors grown on electrospan nanofibers and detecting the reporting exosomes released by the tumor cells. An external transceiver patch based on the skull, where the radiofrequency transmitter is located, and a central unit with long-range communication, wireless photo area network integration, localization algorithms, and analysis and intelligent feedback algorithms. The enabling patch and interfaces, which include the radiation frequency mediated reprogramming and external control, converge to an emerging supradiscipline we now call radiofrequency genetics. We had optogenetics, and now we built a new area, the radiofrequency genetics, and established brain molecule, not just brain molecule machine interfaces. This slide summarizes preliminary achievements of Gladiator and address gaps and challenges in the molecular communication field that we have already discussed. Remember the slide with the challenges we had to face. I think we have met some of them. Partners at the University of Ulu and Fraunhofer, in interaction with my team at EBOS, constructed the basic structural units, genetic units, for monitoring and therapeutic bio-nanomachines. And this is a representative figure of glioblastoma release exosomes, monitoring exosomes, interfacing with the neural stem cells. Osaka University, jointly with EBOS team, built the already presented growing bio-nanomachine and glioblastoma evolution simulation model. Waterford Institute of Technology, together with National University of Technology of Norway, developed and already published a controlled therapy simulation model based on the release rate of therapeutic exosomes. 3D tumor construct of glioblastoma cells grown on hybrid nanofiber and hydrogel scaffolds by our team at EBOS comprise the so-called biomimetic molecular communication and biomed. Significant advances have come in and out messaging interfaces, and they are already available by the University of Cyprus and Fraunhofer engineering teams, and include the design and preliminary prototype of implantable optoelectronics, miniaturization techniques for antenna design, which employ biomedical materials in order to reduce the size of the arrays to practical sizes for head placement, and for evaluation of energy absorption inside the body. The optimized design is evaluated using a realistic three-dimensional model, including all the parts of the human head. And this work is complemented by screening for new genes under radiofrequency control jointly with EBOS. So, are we at the dawn of an industrial revolution? I think so. These advancements seem to be bringing us at the real dawn of a new era, a brave new world. Why? Because ongoing collaborative research is taking molecular communication to a drastically new level. An important allusion to the potential of molecular concepts is exemplified by their role as the main technology for implementing advanced medical solutions. Novel strategies are emerging, enabling integration of diagnosis and therapy across major specialties and subspecialties of clinical practice. We have shown that we have highly interdisciplinary research integration, converging to solutions for realizing nanoscale communications. Bio-Nanomachine Diagnostics and Nanonetwork Therapeutics emerge as a superdiscipline, leading to a true paradigm shifting in oncology. Specific advancements across disciplines are addressing predefined challenges and gaps because we have standardization of architecture and simulation framework. Great steps are already taken by the Circle CSA toolbox, and further opportunities for coordinate actions can be provided by schemes such as the COST action. Structural tumor units are now recognized as working prototype models for higher level functionalities. The go and grow paradigm in tumor evolution and the attractive, interactive exosome uptake release loops epitomize what we define as collective bio-nanomachine behavior. What else? Three-dimensional bio-inspired organoids and tumor constructs define biosimilar molecular communication models, taking and exploiting useful lessons from biology. Engineer bio-nanomachine functions under in-messaging, out-messaging control of optical and radiofrequency interfaces are realizing the Internet of Bio-Nanothings and are bringing the brain-molecule-machine interfaces at a radically new level where computing systems do detect, do localize, and do control molecular activities bidirectionally. And importantly enough, all this can be achieved with minimum human intervention, paving the way to radically new brain-computer connectivity approaches. The challenge for us, the clinicians, remain to build the necessary innovation ecosystem by attracting the clinical community and to stimulate translation to clinical practice. We need to revisit the regulatory environment and, most importantly, reform our higher education and training towards the new generation of molecular communication leaders. My driving force as a physician has always been that no matter how innovative and disruptive our work may be, this innovation should not be for the sake of the technology, should not be for us, the healthcare professionals, but for people's life and their future. And the ultimate goal is to transform all our discoveries to recoveries. The appreciation and funding of the Gladiator project by the European Innovation Council is nothing else but a vote of confidence for the whole molecular communication community, for all of you. The challenge ahead is explicitly defined by those who have evaluated this work, by the reviewers of this project, who quoted that this interdisciplinary project will create significant added value for participants in society. Project results will provide the keys to a complete new regime of fighting deadly diseases. So Gladiator will pave the way to new treatments of cancer, as was the discovery of antibiotics past surgery. And I think this is now becoming our new promise. Certainly no man is an island, and great visions need joint efforts. I therefore want to thank my partners at the Gladiator project, and I'm sure you agree there can be no creation without recreation. My extended team at EPOS and University of Cyprus for contribution to ongoing and research projects that have nourished this work. but I feel obliged to specially mention 3 bio colleagues. Associate Professor Ioannis Krikidis at the University of Cyprus, who introduced me to this mostly exciting field of wireless and molecular communication that has radically transformed my research prospects. Professor Kostas Pitris, a long-standing collaborator who has been the engineering mind in most of my projects, and ultimately a wonderful leader in molecular communication, Professor Tadashi Nakano, for his kind invitation that has given me this unique opportunity to share with you today my fantastic voyage through Bio-nanomachines and nanonetworks. Thank you all for your attention.